Good afternoon. My name is Gary Fortin, and I'm the Chief Operating Officer of Shape Therapeutics. I wanted to start off by thanking the organizers of the meeting for a fantastic meeting so far and for the invitation to present today. Just a quick legal disclaimer that I will be giving some forward-looking statements during the presentation. So Shape Therapeutics was founded 5 years ago in 2018. And it's headquartered in the heart of downtown Seattle. At Shape, we are committed to expanding the potential of RNA therapeutics. At Shape, we harness AI to rewrite the digital code of RNA with our RNA Fix technology. By combining generative AI, the body's own RNA editing machinery, and massive biological data sets, we intend to create breakthroughs in safe, effective, and accessible RNA editing medicines. RNA Fix is an RNA editing technology that leverages natural enzymes in the cell called ADAR. ADAR stands for adenosine deaminases acting on RNA. These endogenous proteins convert RNA adenosine to inosine, which is then recognized by the cellular machinery as guanosine, making a functional A to G base edit. RNA Fix functions by delivering DNA-encoded guide RNAs via AAV. These guide RNAs have sequence complementarity to a target site in the transcriptome, and upon hybridizing with that complementary sequence, recruit ADAR to that specific site for precise and programmable RNA editing. This powerful tool has immense potential, correcting disease-causing mutations, modulating gene expression, even altering protein function. And it does so with some key advantages over alternative editing mechanisms. For starters, RNA Fix has been shown to provide efficient editing in all cell types tested, including terminally differentiated neurons and muscle cells. Unlike DNA editing, RNA editing avoids permanent genetic modifications, as seen with CRISPR, base editing, or prime editing. And unlike some DNA editing tools, RNA Fix does not rely on the introduction of big protein, potentially immunogenic protein complexes to edit the RNA. It relies, like I said, on those DNA-encoded guide RNAs that are maintained in cells in endosomes, providing the potential for durable one-time treatment. The biggest challenges to consider when designing ADAR recruiting guide RNAs are, one, ADAR's natural sequence preference, and two, that ADAR is innately promiscuous enzyme. So on the left, I have the natural sequence preference for ADAR, and as you can see, ADAR actually disfavors adenosines that have a 5' guanosine neighbor. A catalog of 5,000 pathogenic G-to-A mutations shows that roughly a third of those adenosines actually have a 5' guanosine neighbor. On the right is evidence of ADAR's promiscuous nature. So here we designed a canonical guide RNA with a single mismatch to an adenosine in the Rab7A gene. RNA-seq analysis revealed that 45% of the target adenosine was edited. However, there is promiscuity across the transcript with a lot of off-target adenosines impacted as well. So how do we design guide RNAs that overcome ADAR sequence preference and this promiscuity? Our AI-powered guide RNA discovery platform enables us to overcome these challenges. Over the past 5 years, Shape has generated massive guide RNA data sets across thousands of genetic targets, incorporating immense sequence and structural diversity, each with their own unique editing signature along the RNA. Using these data sets, our AI engine trains on the rules governing guide RNA-mediated ADAR efficiency and specificity. These rules can then be employed to generate guide RNAs with novel structures that are optimized for human disease and have higher editing efficiency and greater specificity. Through this generative AI approach, we are confident that we can build a guide RNA for any target. LRRK2 G2019S is considered a key genetic determinant of Parkinson's disease. In this slide, we show the evolution of editing efficiency and specificity for this mutation, first at the top with a canonical guide RNA with full sequence complementarity with that single AC mismatch. You'll see roughly 45% editing, but with significant off-target editing as well. In the middle is a guide RNA identified through high-throughput screening. You have more efficiency at almost 100% editing, but still have that off-target editing across the transcript. And finally, at the bottom is highly efficient and specific editing with an AI-generated guide RNA. With AI-generated guides, we see high-efficiency RNA editing both in vitro and in vivo. Here we show for an undisclosed CNS target how 80% RNA editing when AAV-encoded guide RNAs are delivered to 3 neuronal cell types on the left translates to up to 75% editing in the brains of adult mice. As mentioned briefly earlier, the potential therapeutic application for this technology is extensive, including inhibiting protein translation, correcting pathogenic G to A mutations, modifying disease pathways and protein-protein interactions, and altering RNA splicing and stability. But the true potential of any in vivo genomic medicine sometimes relies on more than the payload. In addition to our RNA-fix platform, the scientists at Shape are applying AI to tackle other key challenges in gene therapy, namely the liver tropism of AAV and the low yield seen with standard AAV manufacturing platforms. Our AAV ID platform is built to deliver tissue-specific AAV, enabling high-precision delivery of therapeutic payloads to non-liver cells. These AAV should not only reduce off-target toxicity, but should enable lower dosing and lower manufacturing burden. Starting with 100 billion unique capsid variants, 1 billion were selected for their ability to assemble virus. These 1 billion variants were administered directly to NHP prior to tissue recovery and sequencing. Sequencing data representing greater than 32 million capsids across 48 tissues were used to train AI models on the rules governing tissue tropism. These rules were then used to generate lead AAV ID capsids for testing and validation. TrueStable is our stably genome-integrated AAV-producing cell line with high-producing clones and highly reproducible product profiles. High yield and high purity will enable reduced dose, reduced toxicity, and reduced cost. Summarizing what the team has developed, the 3 elements required for AAV production are stably integrated into the genome of a parental cell line, creating a stable cell pool with toxic elements silenced. Single cells can then be grown into monoclonal cell lines and screened to identify the best clone with the most homogeneous viral product at the highest titers. At Shape, we've been very lucky to partner with key pharma companies in the field. Earlier this year, we announced our collaboration with Otsuka, developing therapies for the eye, and have been advancing work in Alzheimer's and Parkinson's disease with Roche since 2021. In closing, I'd like to invite you all to visit our new headquarters in downtown Seattle. We moved into the facility in May, and we would love to introduce you to some of the scientists that are doing this work and hear firsthand from our 150 employees why Shape is such a great place to work. So with that, I will, I guess, take questions or not. Thank you.